get started then. Thanks for coming to another rendition of our Protein Design and Modeling Club. Generously sponsored this month by our friends at Gens, who provided all the nice drinks and food. Yes, okay, so while Ava is, while Chris is going to help Ava get set up, I will just mention a few things. So some of you may know Ava because she's been in Boston for quite some time. She has done her undergrad at MIT, where she focused in computer science and molecular biology. From there, she came to Harvard, actually to HMS, to do her PhD in the biophysics program. Is anybody in biophysics here? Okay, no one's in biophysics here. But some people might be watching in biophysics. So Ava is a proud biophysics Harvard alum. And directly from there, she went over to Microsoft, to MSR, and has been there for a little bit over 2 years now, doing some pretty cool stuff that she is going to share with us tonight. So welcome, Ava, and thanks so much.
All right. Is that working? Test. Okay, good. Okay, well, thank you, everyone. Thank you, Chris, Nick, and Sergey for the invitation and for having me. Pleasure to have this time to share with you some of our recent work from our team at MSR, where we've been developing new generative models for protein design, specifically trying to root our approaches in the fundamental biology and biophysics of protein sequence and structure, and using that to inform the types of models that we design and the types of settings that we use them in. So to start, I want to take a step way back and think about really what is the nature of the problem that we're talking about when we think about generative methods for biology at large. And so this image that I'm showing you here is a scientifically to scale and accurate artist's rendition of the nucleus in a cell at 1 million times magnification. And what you're seeing here is, if you can appreciate the nuclear membrane, a pore complex in that membrane. And why I like to show this image is really to remind us of the underlying complexity of biological systems when we get down to the molecular level. So even when we think about designing new protein sequences or protein structures using computational methods like generative models, I think it always helps to contextualize the setting that we're really thinking about and talking about when we want to move some of these methods into the real biological world. And furthermore, my ultimate goal as a scientist working at this intersection of computation and biology is really to derive new biological insight and understanding. And when we think about, okay, why generative modeling, why design, from that perspective, there's a lot of angles we can think about it. But one more philosophical note that I want to share is this beautiful quote from Richard Feynman, who stated, "What I cannot create, I cannot understand." And I think this goes both ways. And hopefully the idea is that by deploying some of these protein design methods, we can get more insight into the underlying biology of these amazing biomolecules and vice versa. And so with that, as a final broader note before diving deeper, this is really the goal of our team all up at Microsoft Research, where we have a team specifically focused on developing new machine learning and AI methods for problems at all scales of biology and data sets of all types of resolution, all the way down from the molecular level, all the way up to the patient level, thinking about disease modeling, genetic studies, and trying to design new therapies and diagnostics. And so today, for the purpose of this talk in our group, we're going to really focus on some of our recent efforts in the protein engineering realm. And so to that end, I think for this audience, it goes without saying that protein design is of tremendous interest and importance because of this possibility to now leverage de novo design proteins to unlock new molecular, therapeutic, and biological capabilities. And the goal here is to leverage design proteins to try to expand the scope of functional space that these molecules can access. For everything from new molecular tools like GFP, shown here on the left, to enzymes that can perform new chemistry, for example, the catalysis of carbon-silicon bonds, reactions that are not typically seen in nature, to ultimately the design of new therapeutics, such as the enzyme asparaginase, which is used as a clinically approved chemotherapy in certain leukemias. And so across all these examples, the ultimate question, right, is how can we design new proteins to try to expand this functional space? And the goal of generative modeling to that end is to leverage natural data from biology to try to build computational models that can assist us in this quest. And ultimately, as I kind of motivated at the beginning, I think the vision is that we can leverage these models to go the other way as well, to help us find something interesting and new about biology that maybe we didn't know before. And so the real thesis of today's talk is that we're going to be considering the protein design problem across scales, starting with structure and also moving to sequence, and motivated by this vision of trying to develop new generative methods that are biologically grounded to try to guide this protein design problem. And so for that, I'm going to start in the middle of this paradigm of sequence structure to function with some of our recent work in developing new generative models of protein structures. And so going along with this theme, we really wanted to draw inspiration from some of the native biophysics of protein folding and protein structural biology. In fact, we thought about a little bit, okay, how do proteins actually fold to adopt a final structure? As a protein is being made, if we think of this at a very high level, during translation, perhaps initially it can adopt an unfolded configuration, which let's say corresponds to an effectively random configuration of angles in this 3D space. Now, during the folding process, the chain is going to twist and sample different molecular conformations to eventually arrive at a final folded 3D structure. And so considering this, we realized and raised the observation that, well, the bonds that connect the atoms in this protein chain are completely defined by a set of angles, which can be denoted on this diagram here. And if we think about now a completely unfolded random structure, let's assume that those angles can adopt a completely random geometry within some constraints. And then ultimately, when we arrive at a final folded configuration, there's a highly ordered structure to this set of angles that will then denote and specify the overall structure of the protein because of the fact that it's adopting a particular 3D conformation. And so looking at this connection between the possibility of going from this noisy random configuration of bond angles to a final set of angles that specify a 3D structure, we raised the question of, is there a way that we can learn a generative model that can translate between these two of a set of completely random angles to design new sets of bond angles that would specify a protein's 3D conformation? And so this was the question that a former intern in our group at MSR, very talented PhD student Kevin Wu, raised. And he reasoned and posited that we could build a new generative model, a diffusion-based model of protein structure that could operate and translate between these two extremes of a random set of bond angles to eventually design new proteins as specified by their bond angles. And so what I'm showing here is now a visualization of samples from that generative model as it is performing a denoising process of actually moving from a set of random angles to a final set that specify a 3D geometry and structure. And so we're going to get into a little bit about the details of our model and our method and a taste of the results before moving on further. And because of this natural inspiration we draw from the native biophysics of the protein folding process, we call our method folding diff, for folding diffusion for short. And the principle here is that we have built a diffusion-based generative model where the concept is we can start with sets of natural bond angles, right, from naturally occurring proteins, and constitute a set of iteratively noised or iteratively corrupted sets of bond angles by incrementally adding noise in a controlled corruption manner. And so the principle here is that those samples across these individual time steps will be constructed such that at the final time step, we end up with sets of random bond angles that specify a completely unfolded geometry. Then to actually build our generative model for protein structure design, we can learn a neural network that can be parameterized quite simply to basically learn how to undo this corruption process, reversing this noising to eventually be able to generate realistic structures at inference time.
When you do the noising, do you allow for steric, do you allow for collisions?
Yeah, so the question is if during the noising, do we do any explicit accounting for potential steric collisions or clashes? The direct answer is no. The only accountants for the geometry that we do is we account for the periodicity of the angles. So it's not a complete Gaussian noising. Instead, we acknowledge that the angles are going to be occupying a range, so like negative pi to pi if it's in radians, and account for this in the corruption scheme. And we later did an analysis to assess the occurrence of steric clashes in generations from our model.
Maybe you'll get to this in another slide. So if you are, no worries. But I'm trying to understand the structure of your matrices here. So you have what seems to be three angles, phi, psi, omega. But then I see more than, I assume every column is a residue in the protein.
Mm-hmm.
But I see more than 3 rows. Are you discretizing the angles and you have bins for them, or?
Yeah.
I'm trying to understand what these mean.
Yeah, so it's a little small here, but we have the set of angles is 6 dihedrals and torsion angles. Yeah. So phi, psi, omega, and these 3 theta angles that are.
Oh, and so the theta angles are always so close, always so similar. That's why they're all.
Yeah.
Did it actually help to add that part of the noise?
Yeah, we did an ablation of this, and we found that most of the information is in the phi, psi, and omega. But
What I'm wondering is, does it add some extra degrees of freedom that helps during the diffusion?
in terms of incorporate.
So let's say if you were to fix the angles to their ideal more.
I see.
And not allow those to change during the diffusion.
I see. I don't think we did this exact analysis, but yeah, that's a good suggestion. Yeah. Okay, so one advantage of using this angular formulation for the structured design problem is the fact that the network that we use to actually learn this denoising process can be quite simple. So in our case, we use a plain vanilla transformer because of the fact that the equivariance requirements of the geometric problem, where the protein has to be invariant around rotation and translation, are inherently baked into the fact that we're using this angular representation. And so what that means is that this network does not have to be a complex equivariant an architecture. It can be something quite simple because all those constraints are inherently represented within this angular representation itself. And so to contextualize that, I have that kind of visualized here, where with this, our folding diffusion approach, we're learning over these internal angle sets which contrasts to common prior methods in structure-based diffusion models for structure design that rely on 3D coordinate representations of the angle of the protein structure. And so in the context of a 3D coordinate system, right, each of the individual atoms are specified by XYZ coordinates in 3D space, which means that when you learn a diffusion-based model, you have to incorporate these requirements to preserve invariance under rotation and such into the work architecture itself. And so that's one modeling-based complementary aspect of our modeling approach compared to methods that rely on 3D coordinates. And since there have been other approaches as well by thinking about positional frames and so on, but for now, I'm just highlighting the relationship between these two. And so I'll give you a very brief taste of some of the evaluations that we did with our folding diff approach to assess the quality of the structures generated, and then move forward into some of our more recent work. And primarily, we consider three axes of evaluation for structures that we generated from diff. Firstly, whether we could even recapitulate the distribution of angles in these structures, whether we could capture known structural motifs and structural elements, and finally, what was the quality and designability of the protein structures overall. And so firstly, thinking about the distributions of angles that were captured in these generations, we compared the fact that in true proteins, right, these angles are not independently distributed. They exhibit very strong correlations. And so if we visualize the relationship between phi and psi on these Ramachandran plots, we see that there are defined regions of the space that correspond to key structural motifs. And indeed, when we do this similar analysis on generations from folding diff, we can see that we can recapitulate those same patterns of protein backbone angles that define these key structural motifs. We can also look at the structures overall qualitatively. So this is just a sample of a few picked examples of generations from folding diff compared to the most structurally similar structures from the natural set. And qualitatively here, we're just seeing that we are getting distinct structures, but similar patterns and motifs being exhibited. And I'm not going to go further into the suite of quantitative analyses that we did for the purpose of time, but we also assessed the relative diversity of these backbone structures generated and how designable they were in the sense of, could we realize a native amino acid sequence that could specify that 3D geometries specified by those angle sets. So this is for now all I'm going to say about the structure-based problem. But I will note that, since we put the folding diffusion work out, there have been a flurry of works that have approached the protein design problem from the lens of structure-based design and using diffusion models as a toolkit for solving this problem. Notably, RF diffusion, chroma, similar works that were shortly following our preprint on folding diffusion. Yeah.
Before you move on from this, one of the maybe main hurdles, I guess, for doing a strictly angular-based approach would be the so-called lever arm effect. You probably get asked this question a ton. So,
yeah.
How does the model do? Essentially, you have a small error in the middle of the protein, by your side, and now the whole rest of the protein is thrown off. So does it correct itself somehow? Is it a transformer? Or?
Absolutely, yeah. So the question is about the lever arm effect, and we did an analysis of this. We showed that indeed, within the middle of the protein, those residues, if there are small perturbations in the angles, there is a propagated effect on the structure overall. But for the scale of perturbations that we would expect, that we assessed, okay, what is the structural similarity of if we put in a perturbation in those internal positions relative to the structure without any perturbations, and we can still see a decently high structural similarity between the two. So there is some effect. It's not super, super huge. I don't have the plots of this, but they should be coming out in the full version of our paper, which is soon to be published. Yeah.
Someone online is complaining that the background noise is like on. There's background noise, Michael, is too sensitive or something.
My mic. It could be my hair.
It's better to.
I could put my hair up and see if that corrects it. It's a very real problem. Okay, well, we'll see, and they can specify, yeah. Okay, so I talked a little bit about structure, right? But in truth, you know, if we think about this paradigm, this traditional paradigm of sequence to structure to function, we raise the question and the hypothesis that structure is this middleman, right, in this process. What if we could learn directly from sequence and employ this generative diffusion-based paradigm to generate directly over sequence space? And our rationale and reasoning for that was the fact that we saw sequence as a universal and foundational design space for proteins, in that it is the encoding of the natural language of protein biology. Motivation for this concretely comes down to a few different factors. The first being that with any structure-based method, there's this requirement to do this indirect sequence design step if you're actually going to try to, you know, make that protein and realize that protein in real life, right? So given a structure, you have to inverse fold and infer an amino acid sequence that's likely to adopt that final structure. The second being that sequence serves as a very useful space because of the fact that there are these large-scale unannotated and annotated data sets. And as a result, there have been a number of function predictors that have been trained on these data sets of primary amino acid sequence to now predict and score these sequences for a function. Underlying all that is the fact that in the information that we have available, the scope of sequence data available far, far outweighs the scope of characterized structural data available. So if we think about the whole PDB, right, this is about 200,000 solved crystal structures of unique proteins, whereas a data set like UniRef50 has on the order of 50 million unique amino acid sequences. And finally, because of the fact, another fact is that there are some regions of protein functional space that are traditionally inaccessible to structure-based methods, namely with proteins that have intrinsically disordered regions or that are intrinsically disordered. We reasoned that by learning over sequence space, this would allow us greater flexibility to model and capture this rich aspect of biology as well. And so we thought about taking this paradigm, taking our diffusion-based modeling approach and lens, and trying to leverage sequence as a universal design space to design new generative models that would be controllable and programmable by way of leveraging new breakthroughs in diffusion-based modeling. And so this is work that was very much spearheaded by a data scientist on our team, Sarah Alamdari, that we recently pre-printed about a month or so ago. And we have developed a model that we call EvoDiff for evolutionary-scale diffusion that is a generative diffusion model that learns and operates over amino acid sequence space for protein sequence generation. And the concept of this diffusion modeling paradigm, where we have this iterative corruption or iterative noising, and learning a neural network to undo that noising and then be able to generate new instances at inference, is exactly what we employ in this framework. The key difference, though, is that now, instead of thinking about a representation of structure, we're learning and operating directly over amino acid sequences. And so, to walk you through the couple of frameworks that we developed to do this, the first framework that we developed and employed is this framework that we call order agnostic autoregressive diffusion, where basically the corruption scheme comes down to the introduction of masking tokens, specifically masking tokens in this iterative process. And importantly, this stepwise corruption can occur in any arbitrary order, in any position in the amino acid sequence. And so starting from native sequences, we iteratively add these masking tokens as a way of corrupting, to the point where at the final time step, we have an all-mask sequence. Then the neural network is trained to learn how to undo this corruption stepwise, to then be able to eventually generate new sequences. And so, looking at this visualization here on the bottom right, a particular residue or position in the sequence chosen at random is masked. This is done stepwise in this corruption scheme. And then the neural network is trained to predict the identity of that masked token at that position. This set of masked positions can be done in any arbitrary order, such that the network learns to decode all possible orders of this masking process. And so then, when we now have our trained model that's been trained to remove this corruption, at inference, meaning at sampling time, we can begin with an all-mask sequence and decode a new sequence to generate and to produce a new generation by starting from all-mask. Yes?
So I guess when I think of noise or corruption, I would assume you're mutating amino acids. Can you say maybe why masking versus randomly changing?
Yes, absolutely. So the question is about mutating versus masking, and it transitions exactly to the next slide. So we also thought about doing this corruption where we mutate the individual amino acid identities. And indeed, this was a second scheme that we directly developed and tested, where now we can specify a transition matrix of probabilities of likelihood of any one amino acid to mutate to another, whether that's uniform or informed by some biological prior, like Blossom substitution matrices. And the concept here is now your corruption is this stepwise mutation, and the network learns to undo that corruption. Go ahead.
You are undoing the corruption. You're feeding it the positions or the phi psi.
So now we have transitioned away from structure completely. So this now all it's seeing is the identities of the amino acid residues at particular positions in the sequence.
But no information.
No structural information. This is purely sequence level information. And so to that question about how does this mutation scheme compare to the masking scheme, what we observed empirically was that the masking-based strategy far outperforms the mutation-based corruption. And our loose intuition for this is that because it's seeing a particular masked token, and that's denoted by the special token identity, perhaps it is an easier task for the network in terms of knowing which positions are corrupted. Whereas in the mutation scheme, it's just another amino acid. So potentially there is greater capacity to be learned here, but at the cost of a harder learning task.
Maybe my question is very basic, but if there's a deletion and insertion kind of complex variation happening, can you model that?
Yeah, so in this case, the task is to just generate a single protein amino acid sequence. And so within that context of one protein amino acid sequence, the insertion and deletion is usually thought about relative to, let's say, an alignment of related sequences. In this case that I'm showing here, we're just considering one protein amino acid sequence and generating of a specified length. And so if that addresses it, yeah.
I'm just kind of comparing it to like some of the other diffusion models where there's like a fixed number of steps that you need to like take something fully to noise.
Yeah.
And here it seems like the number of steps would depend on like the length of the sequence. Is that how you do it, or is there some other way?
Right. So in this case, we set a particular number of steps overall, but most of all the protein sequences are of shorter length than. So here the corruption is matching the number of steps required to get to en masse, if that makes sense.
Does that end up leading to like a size limit, like the length of the sequences we can sample?
Yeah, so with these sequence-based models, we went up to sequences of 2048 amino acids in length. Did not test beyond that, which is pretty long and gives us pretty good coverage. There are also ways in which with like this mutation-based strategy, this type of corruption, you can control basically the schedule of noise that's added so that you can bias like more heavy mutations up front or later in the diffusion process relative to the number of time steps as a whole. Yeah.
There's some online questions. So Amir is asking, could you share some details around the neural network architecture search?
In terms of I'm guessing the underlying question is what is the neural network that is used to actually learn this model? And so I'll get to this slide here. Yeah, so as we kind of were discussing, we had these two schemes. We found that the masking-based scheme significantly outperformed the mutation-based scheme. To the question specifically about what is the underlying neural network architecture, in our case, we have a convolution-based architecture that's based on a ByteNet style. So if you're familiar with the paper CARP, which is led by my colleague Kevin Yang, who's also on this paper, we adopt a similar architecture for the neural network that's actually learned to build our EvoDiff model. And importantly, right, in addition to this diffusion-based framework, another key aspect of our modeling approach was the fact that we leveraged an evolutionary scale data set to actually build this model. And this was the first such instance of a diffusion model of protein sequence that was trained on evolutionary scale sequence data amounting to approximately 50 million unique sequences from UniRef 50. And so that gives us kind of an overview of the modeling approach that we took here. And now to transition to thinking more about the results in terms of the generations from this model and how we are able to evaluate it, to really contextualize and show concretely how this generation process works. In this masking strategy, we can start from a sequence of defined length, all masked tokens. And what the EvoDiff model does at inference time is basically stepwise undo this corruption, starting from all mask. And what I'm showing you here on the left is a visualization of that sequence-based generation, that stepwise generation. And what you see here on the right is a pure visualization of the likely predicted structure from that sequence. And so importantly to the prior question, our model is solely operating over sequence space, and any structure visualizations I show are, after the fact, not direct generations from the model itself. So we combine these two key aspects to our modeling framework, the first being this evolutionary scale data and training, and secondly, this discrete diffusion approach with this masking strategy. And to go through our core empirical results, we really were considering three axes. First, whether or not these generations were plausible structurally and in terms of the quality of these generations, how diverse they were in terms of similarity to known proteins and coverage of protein functional and structural space. And finally, one of the key advantages of a diffusion-based approach is that it gives us a framework for thinking about how to guide or condition generation according to specific constraints. And so I'll show some early results in our first attempts to do that. So first, thinking about purely the plausibility of these individual generations from our model, we developed a workflow to assess this, where we take our EvoDiff diffusion model, sample from it to generate a set of protein sequences, and then use a structure prediction tool, external tool like AlphaFold or OmegaFold, to predict a likely structure, assess its confidence. And then we can perform this auxiliary step of inverse folding a likely amino acid sequence that is likely to fold into that structure. And along this evaluation workflow, we can consider what is the structural confidence of the predicted structure, and what is the self-consistency and similarity of this inverse folded sequence relative to our original generation. And so if we compare these two metrics of that confidence in structural prediction versus that self-consistency, we see that first, for natural proteins from the test set, there's a good distribution along this axis, and that generations from our EvoDiff model occupy this range with a good fraction of these generations exhibiting high structural confidence and high self-consistency, indicative of greater likely plausibility. We can also visualize some of these generations individually in terms of the predicted structures of these designs, and characterize, what is the confidence associated with the structural prediction and their similarity to sequences in the test distribution. One key aspect that we wanted to understand to contextualize the performance of our model was how well this evolutionary scale diffusion approach compares relative to other approaches for protein generation. Yes.
Sorry, one quick question maybe on
Yeah.
the last slide. So if I understand the complete de novo generation correctly, starts off fully masked and then starts adding in sequences. The first few residues essentially are likely to be mostly random. Is that a fair assessment?
Yes, so we start from all mask. The position of the first decoded residue is randomly chosen. And so it's everything, there's a great diversity associated and variability inserted by virtue of the fact that you're selecting what position to decode at random. So those first few are likely to be effectively but keep in mind that the model is trained on all of this data to learn the pattern learn a representation of the data that's present. And so that will be, I guess, yeah.
So do the first few random sequences, amino acids that get chosen, effectively define what your ultimate topology is going to be?
I see. So yeah, that's a good question. Yeah, because of this iterative denoising and reversal of the corruption. my intuition is that yes, yeah, you're going to be somewhat constrained, but if these positions are also decoded at random, then it's not the case where you have a set of, let's say, contiguous residues that are decoded on block, right? And so the topology is not as restricted as if you were to, say, decode this portion first. We can also play with, and in fact, I think we did some investigations to this of selecting positions that based on, let's say, conservation and not decoding in a random order, but selecting an order of decoding such that it would be more matched to relative conservation compared to the whole distribution. And so that can also improve in terms of eliminating some generations that are less possible.
Can you introduce maybe more noise by putting masking back in the middle of the demasking process?
I see. So
kick yourself out of a minima and allow it to maybe reach a more optimal overall solution.
And then basically going back to a prior step. Yeah, that could be interesting. We haven't experimented carefully with that.
I'll go first, I guess, because I'm here first. So I'm interested in that. I think this is very interesting, this way of analyzing the model performance with ELDET to predict the structures, but it's sort of a little antithetical, I suppose, to something you said earlier about intrinsically disordered proteins and being able to then capture that. Because I guess I wonder if you take the 50 million sequences in your training set and run a intrinsically disordered predictor on it.
Yeah.
What's the percent of those sequences that have at least long stretches that are IDRs? And then, so I mean, I sort of wonder how much you should expect this to be a valid metric for what you're training on.
Absolutely, I completely agree with that point. It is an imperfect metric, and in fact, we see this or kind of recapitulated where, not everything in the gray points are the test points from the test set of UniRef 50 in our split, and so not everything is occupying this bottom right corner of very high structural confidence, because as you said, the PLDT is an imperfect metric and it's going to be biased towards what is well represented or overlaps with, what was used to train these structure prediction models. So absolutely in agreement with there.
The second part, I guess, sort of corollary to my question was if you took the training sequences, ran an IDR predictor on it, and then filtered those out, trained your model on sequences that are predicted to be structured, would this assessment get better? That's the question.
That's a quite interesting experiment. I think there's also an underlying caveat here, and these IDR prediction tools are not super. And they rely a lot on, I believe, on homology-based information in terms of how those IDR scoring tools were actually built. But that would be an interesting experiment to filter out for what we expect to be structured and see if we can now bias the generations towards occupying this, more structured region of the space. Yeah. It's a great suggestion.
I'll save the questions till the end because they're more general.
Okay, great. So if we compare our diffusion-based approach to now a traditional, like left to right autoregressive approach, where now you have a fixed decoding order, you start with one token, you predict the next amino acid. Based on that, you now have two, you predict the next, and so on in this left to right manner. We see that the left to right model is, and we can quantify this as well, generates slightly more that are biased towards this bottom right region. But importantly, if we now qualitatively compare to a mass language model, the generations from both our EvoDiff model and the left to right autoregressive model are overall occupying more of this downstream diagonal in this visualization. And the fact there is that both of these approaches, the diffusion-based approach and a traditional left to right autoregressive approach, are explicitly trained in a generative task, whereas the mass language model is a single step of predicting mass tokens, whereas the diffusion is multiple steps of decoding. And so importantly, right, it gets at this notion of plausibility, but still, as we kind of were discussing, it's very much through this bias and lens of what has been established with these structure-based tools. We also wanted to consider now across the scope of our generations, what is the coverage over natural functional space and natural structural space and the diversity of these sequences. And so here, the first thing we did was we took sequences from our test split. We leveraged a language model-based tool, Motif, which has been explicitly shown to capture functional annotations in its embeddings, in the sense that embeddings from this model will separate well by known functional annotations. And we used Motif to generate embeddings for these sequences and then visualize them in 2D using UMAP. And so this is what you're seeing here, 2D visualization of these resulting embeddings over the test set. We can do a similar analysis for now taking generations from our EvoDiff model, predicting those embeddings, visualizing via UMAP, and then overlapping the two to see how well generations from EvoDiff are covering this representation of functional space, at least as captured by embeddings from Motif. And what you can appreciate is that we have fairly good coverage, but that there are regions of this space, at least as visualized here, that are not captured by generations from EvoDiff. And this is something that we have yet to look into further about what the nature of these proteins and are in the natural set. We can also measure this quantitatively by computing effectively a distance metric, looking at the similarity of the distribution of embeddings for natural proteins versus generations from EvoDiff, and measure this quantitatively. But ultimately, these numbers and visualizations are without context unless we have something directly comparable to benchmark and assess relative to. So if we look at generations from a left to right autoregressive model, we see that at least by this distance-based metric, the generations from the left to right autoregressive model are distributionally closer to natural proteins in this embedding space. But importantly, our method, by the fact that we're not decoding in a fixed order, gives us greater flexibility to now do different forms of conditioning, as I'll present a little bit later on. Compared to a mass language model approach, we see that both these two generative approaches are vastly better in terms of their coverage of this representation of functional space. And furthermore, compared to a state-of-the-art generations from a state-of-the-art structure-based model like RF diffusion, we see that we have better coverage over this representation as well. Question? Yeah.
For the protein language model, is this like you're starting with all masks and then doing like kind of GIF sampling? How are you generating the sequence?
Yes. For the mass language model, it is GIF sampling. Yes.
For the RF diffusion, are you using protein internet or something?
Yes. For the structure-based method, we took the generated equivalent number of structures from RF diffusion, inverse folded, and then ran the analysis on those inverse folded sequences. I think there was another question. Okay, great, thank you. We can, so that is using a language model that's been shown to capture functional space and functional annotations. We can perform an analogous analysis using a model that classifies residues for their likely structural class or structural element. So here, this is a 3-state structural annotation that's per residue, taking generations from sequences from the validation set, sequences from the generation, classifying based on likely structural feature for each of these residues. And we can also do comparisons quantitatively, looking at the distribution of structural features across. And so first, you're seeing sequences from the validation set, the predicted secondary structure annotations, and what that distribution looks like. We can then compare that alongside to generations from EvoDiff, and you can appreciate that we see very similar distributions, both qualitatively and quantitatively, in the secondary structure space, and again can contextualize relative to those 3 baseline methods of a left to right model, Gibbs sampling from ESM and sequences inverse folded from RF diffusion. And in this bottom right, what you'll appreciate is that there's a very, very strong bias in the structural characteristics of these generations from RF diffusion. And importantly, this bottom left corner that represents a less structured space is not captured at all by RF diffusion, whereas we get coverage of this as is recapitulated in the validation set. So this shows us that our generations can capture structure elements that mirror that distributionally as what we see in natural proteins. And so finally, the last set of results that I'll present is our early efforts in leveraging this framework to do different forms of conditional design. So within this masked framework, the most direct way to do this without injecting any form of external modalities or tools is by leveraging this masking strategy directly to now conditioned design. And the intuition and the approach for doing that is pretty simple. Because the model has been trained to learn this all possible steps and all possible orders of this masking and reversal of that masking, what we can do is within a sequence, rather than masking all of the tokens to start the generation, we can fix a portion, mask the remaining portion, and ask the model to basically now generate only in those masked portions, given the context from the regions that we fix. And what this amounts to then is that we can do this decoding at inference time directly without any additional training of the model, because it has been in its native training learned to do this decoding of all possible orders, arbitrary orders. And importantly, we explored a couple of different strategies for,
Rakesh is asking, can the EvoDiff model generate sequence of variable lengths conditioned on some kind of, so I guess related to the slide you showed, like, could you potentially do variable lengths, maybe add masks or delete masks, I don't know.
Yes, so we can, starting from all mask, you can specify a desired length and then generate accordingly, as well as in the strategy where only a portion is masked, that degree of the number of residues you mask is arbitrary, and you can say have discontinuous subsets of the sequence that are masked. So upfront, some masked portion, and then unmasked, followed by a masked portion. So yeah, so we explored a few different forms of this conditional generation. The first being, and for the sake of time, I can jump back to this if it's of interest, But we also trained similar models that can operate not on a single sequence, but can take in a multiple sequence alignment and generate entire MSAs. And so the conditioning strategy there is that if we are given a multiple sequence alignment, you can mask only a single reference sequence, the query sequence, and leverage the remainder of the information in the MSA as a form of conditioning to now have the model regenerate that individual query reference sequence. And the concept here is that it would allow for generation of proteins that are related, say, of the same family as that has been specified in the MSA. The other forms of conditioning that we explored are more directly related to this subsequence masking strategy, where now, let's say we have a continuous domain that we want to inpaint, we can mask that portion and then ask the model to inpaint and generate that, that region. Or we can do masking of discontinuous sequences. Let's say there's a motif or a functional region that's specified according to a discontinuous stretch of amino acids. Those positions can be specified by the mask token, and then again, the model generates new variants over that. And so I'll briefly mention about the training of this MSA-based model, where the concept is now, given a single sequence, we can construct an MSA by performing homology search to gather related, evolutionarily related sequences. And now, using that same EvoDiff masking-based framework, instead of operating over a single amino acid sequence, the corruption occurs over the entire MSA, and thus we can generate entire MSAs using this approach. And similarly, the concept is we want to leverage a large data set taken from functions and organisms across evolutionary scale, and we can use a backbone model architecture like MSA transformer that's already set up to learn over, this matrix specification of sequences that is an MSA. And I won't go too into depth into the empirical results for time, but the concept here is that by leveraging this explicit generation and learning over MSAs, we can effectively build an amortized local model to generate sequences of a desired protein family. And the rationale here is that if you wanted to generate new instances of a particular subset or family of proteins, you could try first to build a local model where you take those evolutionarily related sequences, train your generative model, sample from it to get new proteins. But instead, because of the fact that we use our diffusion-based approach to train over evolutionary scale space, now we can perform this conditioning strategy just at inference time, where we can take that constructed MSA, now only mask out the query reference sequence. The model sees the information from all the proteins that are related and uses this as context to now generate a new instance given that additional information and context. And so I have more empirical results which we can return back to in discussion, but I want to just give you a flavor of this last set of results in the kind of functional motif design task, where we now can mask arbitrary subsequences of the overall sequence and generate accordingly. And our motivation here was driven by the fact that many functions that proteins perform can fall into this paradigm where there is a functional motif or region of the protein that interacts with a binding partner or another interface, and that functional motif is surrounded by a broader scaffold or structure in the protein overall. And this problem, known as the motif scaffolding problem, has been very traditionally approached by structure-based methods, where the entire 3D geometry and specification of the structure of that motif is specified, and then the goal is to design a new scaffold structure that will preserve the particular geometry of that motif in the new design overall. And so we asked whether or not whether structural information was even necessary to try to tackle this problem, whether we could perform a similar type of motif scaffolding using only sequence information. And the way we approached this was to leverage this masking conditioning, where now in this example that I'm showing here, we have a particular protein of interest, calmodulin, with its functional motif binding site highlighted in green. And so because we know the structure and position of that binding site, we know the amino acid identities corresponding to that functional motif. Therefore, what we can do is we can fix these residues of that functional motif, and using EvoDiff, mask out all the remaining residues. So the model is now only seeing the conserved information of the green motif. Everything else is masked. And now at inference time, the model decodes only those masked portions, resulting in this generated sequence. And so the generation of EvoDiff directly is only in the sequence space on the bottom. The visualization of the overall structure is purely, again, a visualization. But this is the concept of leveraging the amino acid identities of that functional motif, masking the remainder of the sequence, and then leveraging EvoDiff at inference time to generate over the remainder. And importantly, this requires no explicit specification of structure. And we can do these types of scaffolding problems with both our sequence-based model as well as the MSA-based model that has this additional evolutionary context. We can look at the success rates for generations from EvoDiff relative to structure-based methods. And so here we benchmark relative to RF diffusion, the state-of-the-art structure-based generative model, across a set of 17 distinct functional motif scaffolding problems that they benchmark against. And we see that with our MSA-based model in particular, we can perform very competitively in terms of the number of quote unquote solved problems that we have, where our criteria is that that functional motif is preserved within 1 angstrom of RMSD of the motif in the true crystal structure. And what is exciting about this approach, in my view, is the fact that there is no explicit structural information. And so we have a lot more flexibility potentially in how we move this model forward to downstream applications. And so in summary, and I have some more results on different parts of this that we can get into in time, but overall, the vision of EvoDiff is a framework for now leveraging diffusion-based models to perform controllable protein design over sequence space alone by combining the power of evolutionary scale data that's present in these large sequence-based data sets with this diffusion-based framework. We can perform unconditional generation starting from all mass tokens and evaluate the plausibility and diversity of these generations. We can perform scaffolding and inpainting by masking portions of the sequence and keeping the desired motif portions fixed. And ultimately, what we're working towards is the ability now to leverage this framework as a foundation, bringing in different types of modalities, different forms of guidance and conditioning to now move towards the ability to more finely control the generative process. And yeah, it was happy to continue the discussion and jump between slides as well.
All right, thanks, Ava. I have a microphone now, so it'll help with the folks on Zoom for hearing us. So, somebody want to, Jeffrey, let's start.
Yeah, thanks for the talk. I'm just wondering about the conditional generation, just the previous slide. For the case where you have a discontiguous motif, how do you decide what order they're in and how much to put in between? And I guess to extent, like in addition to that, it seemed like some of the motifs you had shown, there was one that was like 3 beta strands. And it was, I think it's a little surprising to me that just from the sequence context of those alone, it's able to recapitulate those. So I guess I'm curious if you guys have any like speculation or thoughts on why that's the case, or
Yes.
yeah.
Yes. So the first part of the question was how we actually perform the generation where in these settings where it's discontinuous. So for the purposes of the experiments we showed here, we tested over a range of overall lengths for the generation. We sample across a set of lengths for a particular problem. We keep the space, the number of amino acids in between these discontinuous segments of the motif fixed for the purpose of these examples. So if in the native sequence, let's say there are 10 amino acids between part A of the motif and part B, for the generation, for now, we said, okay, there are going to be 10 amino acids between part A and part B. But there's flexibility in that you could also toggle the number of amino acids in between. We have yet to perform those evaluations. For the results shown here, it's different overall lengths, fixed length in between components of the motif. In terms of why we are able to do decently well in this test, I think really we see a big gap in the overall performance between the sequence-based model and the MSA-based model. So for example, for this generation here on the right, that generation has the context of the residues of the motif, but it also has an alignment of 64 related sequences that are supplied as additional context to the model.
So it's 63 sequences that are not masked, and then one sequence that has everything else masked.
Let me see, I think I have a. Yeah, I think it was visualized somewhere here. We don't have to go back to the schematic. Yeah. So it's here on the right. So with a single sequence, we mask just those portions. But in the MSA-based model, it sees the alignment of related proteins as additional context. Does that answer the question? I mean, we can get into more the relative performance of the sequence-based and the MSA-based.
My worry is that might just make the task too simple. Like if you were to just take the consensus sequence, probably it's going to be predicted by alphabets.
Yeah, I think that's completely a fair assessment. I think one, and indeed we see, we did assess now with the generations over, let's say, 1,000 samples from the model, what's the relative similarity in sequence space of the generation to the closest sequence in the MSA. And we see we can still get novelty from this, but the structural similarity of the predicted structure is more conserved relative to the MSA and relative to the true crystal structure versus if we were to just use the sequence only model. So indeed, the intuition follows, but importantly, we can still get novel generations that are distinct from existing sequences in the MSA. Yeah.
So one of the questions I had is actually related to Jeffrey's question. Since we're on the subject, I'll pipe in. The functional scaffolding problem is a very interesting one to try to approach by sequence alone. Mostly, I think, because it seems quite unconstrained. I mean, the peptides that are very small can form many different structures with that same sequence. And so, for example, if you took active site residues of an enzyme that were separated by tens of amino acids, hundreds of amino acids in the linear sequence, and those were your motif. You had 5 amino acids or even 6. I imagine that would just get lost in the noise unless you had some other hint of what that structure should be and where those amino acids should be in relation to each other, their relative distances. So it just seems really kind of very challenging thing to try to tackle from sequence alone. It's just so like an EF, but there are some that make sense, like that you could approach. Like, for example, a P loop is one where you combine phosphate if it's a helix and a loop into a strand, and that forms in the absence of any other protein too. So there are definitely some problems where I feel like this is definitely maybe more appropriate than others. And I'm just curious what you think about the eventual scope of this kind of approach.
Yeah, I think that's a great point. I think we're just beginning to think about that. The scaffold problem, I agree, it's pretty unconstrained. And I think part of the reason we jumped to it initially was first from this intrinsic question of can sequence alone, whether that's constrained by MSAs or, learned in the representation of the model in the EvoDiff sequence only model, can we perform somewhat reasonably at this task? And I think that the reason we see that the disparity between the MSA model and the sequence-based model is part of the fact that the MSA information is helping you constrain the problem at large. That being said, I think there has been a fairly restrictive focus on these types of scaffolding problems at large because of the dominance of these structure-based approaches for more guided design problems. And so I think in this next phase of our work, we are trying to think more creatively about what would be other applications and use cases that perhaps are more amenable to sequence-based approaches. And certainly if anyone has thoughts or ideas about potential applications, we'd love to discuss more.
Thanks, Chris.
Awesome. Yeah, exciting work. Thanks for sharing. So my mind immediately goes to my own bias, which is binder design. And so I think everything you showed today was a single chain, and just curious about your thoughts on how you might make this side of approach work with multiple chains.
Yeah, it's a great question. I've started thinking a little bit about this, and you're right, everything we showed was single chain. I think one thing that we could try without any modification would basically be if we can look at multi-chains and treat it as like a pseudo single sequence. And let's say you want to fix one portion of that and generate over the rest. Maybe you can do that where you have a binding pair and you mask one component of the binding pair and you keep the other portion fixed and generate over the mask. I think these would be some very immediate and kind of creative things to try. We're just thoughts, we're starting that exploration.
Yeah, there's actually a lot of questions online, but I don't know if we can get through all of them. But I'll try to summarize at least some of the questions. So one of the questions about the UMaps that you showed, and it at least to me kind of felt like if you want to say something about novelty, like you'd almost want to be outside of those gray dots, but in fact, you're almost saying like it's better to be within those dots. And so one of the questions was like, do any of these approaches maybe sample more in the sparse regions, which is something that we would have, I guess, hoped that such a model would do?
Yeah, that's a great great question. I think so. Yeah, I think it would take to thoroughly assess this, look at, you know, what is the relative density across this distribution. The UMAP visualization is pretty limited, as is the use of a model like Pro-T5 to even generate these embeddings in the first place. One thing that I'm interested in is if we can now, let's say, leverage some form of uncertainty or just a relative density calculation to now bias or guide the samples to more underrepresented or unexplored regions of this space at inference time. But prior to that, I agree, yeah, we need to do a characterization of like, what is the relative occupation of less represented areas of this functional space in terms of density compared to our generations.
Right. And I don't know if there's more questions behind, but another question is, have you actually experimentally tested any of these things are they actually functional?
We are in the midst of that right now. Yeah.
Okay.
Yeah.
Okay, alternate.
Right.
Hi, as people are talking about Evo sequence, I want to go back to Evo structure.
Okay.
So like, I think your main selling point is that you want to try to leverage the biophysics of protein folding. I wonder, like, to me, like, to my understanding of protein folding, it's not complete random. Like, there's N to C dependency, or like, if you want to keep it more complicated, there's a co-translation or folding, things like that. Like, have you guys thought about how to model those? Like, for example, there could be some local folding first, and would that improve?
Yeah. So the question is, have we thought about incorporating more structure about the actual constraints of protein folding into this? Yeah. So we, at the moment, our generation is not constrained by any actual biophysical parameters or equations other than the, learning from these structured angle sets. When I say, or when we write that, it takes inspiration from the native folding process, we mean that at a pretty high level, in the sense of thinking about bond angles as like the determinants of the structural process, but not explicitly accounting for some of the constraints, N to C or other biophysical parameters in the modeling itself. I think it would be an interesting thing to think about. Another angle to that is if we can leverage such an approach for sampling different conformations or ensembles, and how that relates to the relative constraints of these angle sets. But at the moment, it's not explicitly accounted for. Yeah.
I see.
Have you considered including that predicted functional data, any predicted functional data in your training data?
We have started to think about this, right? Because with these diffusion models, advantage is you can use external classifiers, for example, to supply guidance. And so now we're beginning these efforts as well. That was a big motivation behind modeling and learning over sequence space compared to structure as well.
Yeah, thanks.
Thanks. Okay, so I'm going to be annoying. I have like a two-part question. The first question is about like contact order. Did you show contact order for the predicted structures?
I did not know.
Okay, so I'm just curious, like, if you look at the predicted structures of the novel generations that don't have high homology to existing sequences, do you see like lots of long range contacts, or is it more like local interactions? And I'm wondering that just because something Chris brought up earlier is that like you're picking early on, you're kind of like locking in certain amino acids and maybe they won't play nice. You kind of don't know how they're going to end up interacting. So maybe that would be a limitation that you'd expect.
It's a fantastic suggestion. We have not done this analysis.
Okay.
Yeah, that's
Yeah.
great question. Yeah,
That would be,
and great suggestion. Thank you. Yeah.
I would be super interested in seeing that. And then the other thing is related to like diffusion and like, I think when I think about like the benefit of diffusion, I think about like tiny little steps and like, you know, normally those steps are being made across the whole protein all at once instead of like one big step per residue. And like sort of an analogy to the tiny steps where you like just alter coordinates a little bit is like maybe having like a distribution of probabilities of like amino acid types per position. And then like, you know, just modifying that a little bit every step. And that's like similar to some work that like Sergey's explored with collab fold, collab design, et cetera. Did you guys explore that kind of like soft movement through sequence space as opposed to like mutations?
Yeah, so I think if I'm understanding correctly, what you're getting at is whether in the scheme where we mutate the individual residues, if you can control basically the intensity of this change or the rate of this change. Is that correct?
Yeah, like basically move away from like discrete sequence to like allow for probability of different amino acid types, especially at the earlier stages. Like maybe by the end you want like, you know, a real sequence that you can like order and get the DNA for. But like early on, maybe it'd be nice to have it be not constrained to just like discrete sequence.
This is another great suggestion. In this work, our schemes were just this masking scheme and that discretized mutation scheme that I showed with the different transition probabilities. But that another fantastic suggestion. I guess I will use that to plug. We first of all, our team is like very flexible in terms of collaborations, especially with academic groups. So if you all have ideas, applications that you'd be keen to work on, we're very, very open. Secondly, is that we're in the active phase of our internship, PhD research internship recruiting. And so if you are interested in interning with our team and are a PhD student, the applications close December 1. We can also propagate some information about that, or you can email me. And but in general, very, very open to working collaboratively with people.
Hi, I had another question maybe for trying to see different length. Have you tried to like at each step, instead of like unmasking some masked token, to like instead with some probability like insert another masked token, because then you can maybe do this thing with the scaffolding but have different length of like gaps between the parts.
So are you suggesting then at inference that you insert a mask?
Yeah.
Okay. I think this relates to the
This is similar. Yeah.
We to start in these evaluations, we have not yet tested that, where we now inject back during the decoding process additional masked tokens to now adjust the flexibility, like basically give more flexibility or to the generation. We have not tried this. Yeah.
Really cool.
Thank you.
I'll do one.
I guess I'll do one more question. So Rosh suggested that people use molecular dynamics as a way to test structure quality, and they were wondering, have you guys tried doing molecular dynamics? and any of these generated structures?
On these full structures. Yeah, so MD on large proteins is very expensive and hard. We have not done any sort of dynamics characterizations of either the structures from folding diff, the structure-based model, or predicted structures from EvoDiff. I will say that there was a work that took folding diff to generate backbone structures that were used basically as seeds for MD related experiments. This was completely distinct work from our group. It was another research effort, but the concept, I think, of using generations from a method like this to seed MD simulations in a particular space and then go around that is an interesting one as well.
Yeah, the question is roughly asked when I think when you showed that slide with little structures. So I'm wondering if they were thinking like, okay, if you put those into an MD simulation, like will they fall apart or they come together? So
I see, to assess like stability or
yeah, like supposedly if
energetic.
it's a good structure, then I'm guessing if you do MD, it won't just explode or.
I see.
That's my interpretation of the question.
Okay.
Thank you.
I have maybe one last question. It's about the comparison with ESM2. And so from what I understand, ESM2 is trained on 15%, 10% mass tokens, and
yes.
So a lot of context, more parameters, I think, than EvoDiff. And so when you start fixing residues, these motifs, you can fix more and more and more, and at some point you're fixing probably the same amount of residues that's now in the training regime of ESM2. And I'm wondering, is there a crossover in performance then where EvoDiff seems to be better at generating sequences when you have no sequence or you have very little sequence? And that was what it was trained to do. Is ESM2, when you have 10% mass sequences or you have a few mass tokens, a few mass residues, is that superior, or is EvoDiff just a better replacement for this in general?
That's quite an interesting point. I think there's a thing that we have to keep in mind there where ESM was not explicitly trained in this generative task. So to the earlier question, the way we're generating samples from ESM is using a Gibbs-based sampling.
Mm-hmm.
And so even though there was this 15% roughly masking in the training, it's not a direct, I don't know if we can call it a direct comparison in terms of Gibbs sampling on top of that versus a purely generative model. But it would be interesting, I think, to think about how does performance and quality of generations change as a function of what percentage of residues in the sequence are masked, to characterize that more systematically.
All right, thanks. So let's thank Ava again and GenScript for sponsoring. So thank you, Ava.
Thank you.
So stick around, there's plenty of food left, plenty of drinks, and keep bugging Ava, she's here. Don't let her run away. All right, thanks. I remember what we're doing for next time.
Oh, yeah.
Oh, and right, so next month, Soon-Jung Yang is going to be speaking. She's a brilliant MIT student, and she's going to be speaking over at the Harvard main campus next month. So see you then.